Thank you. Hello everybody. This is my control talk for the PhD called Rhythmic Coordination in a Network of Interacting Brain Pathways. I really want to stress out from the beginning that it's an ongoing investigation. So all the results that you are going to see are very, very preliminary. It's more about the framework than the results themselves. And is supervised by Claudio, who is here, and Santiago from Alicante. So we start from the very beginning, from the brain waves. In 1924, Hans Berger, a German psychiatrist, was the first one to detect electric activity from the human brain. It's really funny because he was actually searching for telepathy. He was very into telepathy, and he was searching for signals that they can be transmitted from the one brain to the other. And even though his results are very important, he actually proved the contrary of what he was searching, because the electric signals that you find from the brain are very, very low, and it's impossible to use it for telepathetic reasons. Now, what exactly is the thing that we detect when we put electrodes in the brain? So, more or less, what we detect is synaptic activity in the vicinity of the electrode. But the vicinity of the electrode depends really on what type of electrode you use and the setup. So, probably the most famous ones are the EEG that we put outside of the scalp. This is a very typical casket view that we have. But then we can go deeper into the cortex, for example, the surface of the cortex, or even deeper with the local field potential recordings. And these ones are really inside the brain areas that we care about. But even in that more local recordings, still the best analogy that I had found is that it's like putting one microphone in the middle of a soccer field. Probably you're not going to hear the discussions in the audience, but if a goal happens, you are going to detect that. And exactly this is the type of the oscillations that we find are this kind of very synchronous activity from a lot of neurons. And then the oscillations depend on behavior. They are frequency specific up to a degree. The signals in general are broadband. They are not delta frequencies. And also they depend on the brain area. So here we can see an example. This is EEG. So this guy has these commercial caskets. And then at some point he closes his eyes, and you may notice that the signal that we detect is much more oscillatory. And actually, if you see at the spectrogram here, is around 10 hertz, which is associated with closing the eyes. But in this talk, we're not going to talk about EEG. We are going to talk, and the cortex, we are going to talk about the hippocampal formation, which is deep in the brain. And the hippocampal formation consists of two different parts. The one is the entorhinal cortex, which is with blue in this rat scheme there. And then the hippocampus itself, which is painted in pink. And there are three different, very distinct characteristics of the hippocampus that they don't seem to fit with each other. The first one, in terms of function, the first one is the fact that we have found that it can map the external world during navigation. And the most canonical examples of that are the place cells of the hippocampus and the grid cells of the entorhinal cortex. So for example, here, what we see is just the recordings from one single cell and of a rat that walks inside an environment. And whenever this cell spikes, we put a point inside the environment where the rat was at the time of spiking. And you can see that this simple cell creates a grid of the space, a hexagonal grid of the space, and that's why it's called grid cells, while here, a different cell, which this cell is in the hippocampus, in the CA1 area of the hippocampus, this cell only spikes when the rat is at the vicinity of this specific point. So for this reason, they are called place cells. The other characteristic of the hippocampus is that it's very important for memory formation. So this is a picture of the famous patient HM that he had problems with epilepsy, and his doctor decided to lesion parts of the hippocampus in order to treat that. He treated the epilepsy, but after the lesions, he was not able to form new memories, new episodic memories. So episodic memories are for example, memories of episodes. So what you did yesterday, these memories have a time, they have different people that they take part of this memory, etc. And the last characteristic of the hippocampus in terms of function is the fact that we can find a very important oscillatory behavior at 8 Hz, which is called the theta wave. Now, these three characteristics, they seem very different, but there are some theories that they try to unify them. And the way that they do that is by saying that in the end, navigating into an environment is very similar as remembering an episode of your life, because for navigating, for example, if I want to go from here to the other part of the S07, I have to pass through the door, walk through the corridor. I have to do a specific set of things in a specific sequence in time. And the same applies for a memory. A memory has a beginning, and then I remember what I did, and then we end up at the end of the memory. And the theta wave is very important because it works as a clock that can sequence things together. But this is a theoretical result. Actually, the recordings and the reality is much more messier. And one of the asterisks that we can put in this theoretical framework is that actually the hippocampus is not one thing, and the theta of the hippocampus is not one thing either. So actually, if we go much deeper into how we perform experiments, etc., this is another view of the hippocampus. And the recordings, the way that we do it is by putting electrodes. As you can see here, it's the green thing, which each electrode has a lot of different recording sites. And from each recording site, we get a recording. The hippocampus itself has three main different areas, the dentate gyrus, the CA3, and the CA1. And the problem that we have in the hippocampus and all over the brain is that the signals by themselves are very mixed, because when you record here, the problem is that there is volume propagation from different areas nearby, so you actually record from everywhere. The good thing with the hippocampus, actually, so this would be an example of 1 second activity from this electrode that I'm showing you there. The good thing of the hippocampus is that you can use this independent component analysis in order to demix the signals, and it works very well in the hippocampus. It has been shown. So the way that the independent component analysis works is that you find a linear way of remapping the local field potentials into different components that they try to be as independent as possible. And the nice thing with the hippocampus is that it has been shown that these independent components that they come from a mathematical analysis, they actually reflect the activity of the pathways coming from different parts of the brain or from inside the hippocampus to the specific sites where you are recording. So, for example, this red time signal here, which they call it loconosmolecular in this paper, it really reflects the activity of the entorhinal cortex arriving into this specific area of the hippocampus, which is these red lines that we see here. You can extract multiple independent components that they have a physiological meaning. Here I have 4 of them, but the names, I think that they are not important. The important part is that they reflect activity arriving from a different area to the specific area where you are recording from. Now, the thing is that we have different components that they are also independent, and this thing goes really against the single one clock theta hypothesis that I told you earlier, because if you actually study these components, if you filter them in theta around 8 Hz, and you plot them, as you can see here, different colors are different components, you can notice that sometimes they are very synchronized with each other. As you can see in this epoch, we have always the red peak, the yellow peak followed by the red peak followed by the blue peak, and this thing continues in different theta cycles, while in other times like here, the order is not maintained. And the way that you can quantify the synchronization between the different pathways is by using this metric here. Here is called intercycle phase clustering index, but it's a very famous metric because you will notice that it's extremely similar to the Kuramoto order parameter. It's a very typical metric when you want to measure synchronization. This metric goes to 1 when all the things that you measure have exactly the same phase, while it goes to 0 while they always have different phases in times, independent phases in times. And the thing is that this kind of synchronization changes with behavior, so it's important for the animal. Here we see an example of that. So this is a very typical experiment. It's called the T maze, because the whole point is that you have a rat that it runs through the center arm of the T maze. Let's say that in the beginning it goes to the right. Here is the reward place where it drinks a little bit of water. It goes back up again, and then in the next round it has to go through the opposite side, through the left. So the rat, if it performs well in these tasks, is going to be making the infinite symbol in this experiment. And during the center is considered where the rat takes the decision of whether it wants to go right or left. So the center is cognitively more demanding place than in the left or in the right. And actually, if you compute the overall global synchronization between the sides and the center, you see that during the center, the different theta clocks are more synchronized than during the sides. And here you can see how this looks in a single trajectory. So the introduction was a little bit big, but there are two lines that I want you to remember. The first is that there is no single theta clock in the hippocampus. There are different pathways, and these pathways, they synchronize and desynchronize dynamically depending on what the animal is doing. But I think that there are some things lacking from the previous studies. And one of them is the fact that we have different components that they create a network. And we could use a network view in order to analyze these components. So for example, here, this is also from the previous studies. You see this type of really nice motifs where at some time, two independent components are more synchronized, and then all of them are more synchronized, and this changes. But this kind of analysis lacks from the literature. When you have this type of motifs, you can see when these motifs are correlated with different behaviors, and you can argue why they are in this way. And also, you could try to calculate the directionality of the interaction. So if these two are synchronized here, is it because the red is imposing its oscillation to the yellow, or what exactly is there? And then the other way to expand this work is to expand the interactions network, because actually we know, so in the previous work, everything was in the hippocampus. This is the green structure, but actually we know that there are other structures that they are very implicated into the theta, which is, for example, the entorhinal cortex, which is the orange structure here, and the medial septum that we are not going to talk about that in this talk. So now we are entering into the new part of the talk, which is that we have found one dataset that contains the different structures that I talked about. So this is what the dataset looks like. Here we have 1 second of activity. The first 32 recordings are from the hippocampus. The rest recordings are from the entorhinal cortex. And they also give us from the dataset some labels, which are very helpful in order to locate where exactly the recordings were taken place. So for example, whether it was deep part of the entorhinal cortex or superficial, and some other histologically interesting characteristics from the hippocampus. And also we have behavior. So the behavior is the same as before, it's slightly different the setup. So you can see the rat walking. It's again performing this T maze, so the infinite symbol. The only difference is that instead of having the reward sites here, actually the reward sites are lower. And again, it's drinking water. Okay, so then we start again from the things that we already know. So how to extract the independent components from the hippocampus. Here I have put a little bit more details on the extraction part. So we have the local field potential on the one side, and we want to go in a different space linearly, which is the source space, the independent component space. And we do that using this map. this array. And this, which is called the voltage loadings. The voltage loadings are really interesting and they contain a lot of information because they explain how much each ICA affects the different local field potential sites. And when we plot them, we actually notice that they have these very stereotypical forms. And if you are familiar with them, you can really associate them fast. So for example, this one, the blue one, which peaks, has this peak first. This peak is because there is a source inside the actual network at the Shaffer collateral. Then the second peak, the second peak is associated to the locomotion moleculare, etc. Like if you know a little bit about the anatomy, it's easy to do the association. And then you can use other metrics, like for example, the peak spectra. You notice that there is a really nice peak at 8 Hertz, which is the theta rhythm. And depending on the metric that you use, sometimes the extraction and also the animal that you have, sometimes the extraction is better than before. You want to ask?
How do you know each component responds to different regions?
So I think that online they can't hear the question. So the question of Kaichao was how you know which component corresponds to different regions. Yes, I will do it after. So just by, I mean, these things have been reported in the literature. So we know that they have been reported with a lot of details that if you perform the ICA, when you have something that is like double peak, this is the one associated to the Shaffer collateral, and then the second to peak is the locomotion moleculare, etc. And the way that they have checked it in the experiments is that they extract as I did, but then they also stimulate its pathway, and they see that only the independent component associated to the pathway responds to the stimulation. The rest of them does not respond because they are parallel, let's say. And also, yes, you also know the position. Like afterwards, you can do histology. You can see the slices, and you can notice that this peak is associated to this peak. These things are very well reported in the literature. A lot of experiments.
Let's see the peak of difference.
Yes, espera, take this. Also, if you want to make new questions. But just by the peak locations, the spectra, and other metrics that I didn't show here, you can be more or less sure that the extraction was well and associated the different independent components. But this works mostly in the hippocampus, because if you do it in different cortex, and it has to do with the geometry of the area. If you do it in other cortexes, the independent component is not very reliable in giving you specific pathways that you know anatomically that they exist. We are just very lucky that the hippocampus has this kind of structure where everything is layered, but in other non-layered, I will show you now what happens in different structures. And then we go into the new part, which is the entorhinal cortex. So we know some characteristics of the entorhinal cortex, mostly that it also has a nice theta, a strong theta. But the interesting thing with the entorhinal cortex is that as you start from the highest theta site, which is in the layer 3 of the entorhinal cortex, as you go towards layer 2 and layer 1, there is a phase reversal. So as you can see here, we start at a peak. Then as we go down, it becomes what is called a trough, a bottom. And this thing tells us that probably there is more than one theta source. So we called the layer 1, like there is the theta source at the entorhinal cortex layer 3, and also there is a theta source at layer 1 or layer 2. This is not very clear. And also we know that the entorhinal cortex layer 3 is very coherent with a specific site of the hippocampus. So we can use that in order to extract the two theta pathways that we know that they exist in the entorhinal cortex. So here what we see are the theta coherences between all recording sites of our recordings. This big block is the entorhinal cortex, this big block is the hippocampus, and these are the cross coherences. And each area is very coherent with itself. And here what we see is the phase differences between all the recording sites again and one reference site, which is the red point here. And you will notice that this red point is very close in phase with these points here in the hippocampus, that they are very close to the pyramidal layer. And the more you go towards the other side of the recordings, there is a phase difference that is built up until 90 degrees. So this agrees with with the criteria that I have put there, and we can say that around here we have more entorhinal cortex layer 3, and around here we have more entorhinal cortex layer 2. But we still have the problem of the mixture, because whatever you measure here, it could reach up these recording sites and mess up your recordings. So we tried, and that is for the first time, I believe, in the literature, we tried to use the same independent component analysis algorithm as before, but now for the entorhinal cortex, not the hippocampus. And here you can see the result. The first two pathways, they have higher theta than the rest of them, which is nice because we are expecting 2 theta sources. And the other nice thing is that, as you may notice, the first pathway peaks, the voltage loadings peaks at the end of the recording, so around here, while the second pathway peaks at the beginning of the recording, so around here. So by using this information, we can argue that these are cleaner versions of the sources, which is the entorhinal cortex layer 3 and the entorhinal cortex layer 2. And from now on, we are only using these independent components. So after doing all this pre-processing, we get up to this, which is the basic result. We have 6 different pathways. 2 of them are from the entorhinal cortex and 4 of them are from the hippocampus. And in conjunction to that, we have the behavior. And in the behavior, I mean, we have X and Y, but also is more informative if we look at the velocity, because when the rat is not running is because it's taking the reward, it's drinking water, so this is around here, while when it's running is because it's transversing the T maze. And from here, what we want to do is extract phase, power, whatever else we want in terms of oscillations, and calculate synchronization between all the different pathways, and then relate that to the behavior, go back to the behavior. So here is one example of that. I am showing you phase synchronization using the phase locking value index. Again, is a very similar index to the one that I showed you in the beginning. It looks like the Kuramoto order parameter. We have an exponential and the phase differences between the thing. And I am showing you all different pathway to pathway interaction on top of the behavior. So this is the T maze, the trajectory in the T maze, and you see how much synchronized each pathway with another pathway is depending on the place. It's a little bit heavy, so we can average it all together. And this is a really nice sanity test result, because this is exactly the result that I showed you, which was reported in the previous paper. We have more synchronization in the central part than during the side parts of the T maze. But now here is interesting because we have added 2 more pathways than before. We have added the entorhinal cortex ones, but the result, it remains, it's the same result. Here we can also look at the pathway to pathway, at the pairwise interaction, which was not done in the paper. And here is interesting because you may notice that this column, the first column, seems to be the one which has the highest variations. All the other combinations of pathways, I mean, some of them, they do have some variation, but it's not so big in magnitude. And if you notice, all of them, they have the characteristic that they contain this one specific pathway, which is called the Schaffer collateral. So it seems that the relationship of this pathway with the rest of all the pathways is the one which drives the global dynamics. And finally, so that was the face-to-face relationship. Now we go to the power-to-power relationship. So here the x-axis and the y-axis are just the power of the two different pathways. And again, here I am using this legend, which I'm not sure if you can read it actually, which is an extension of the Pearson correlation, but also for non-linear measures. So it's 1 when they are completely dependent and 0 when are completely independent. And if you look at it with, if you give some time to it, you will notice again that the lowest dependency between two pathways are when one of the pathways again is the Shaffer collateral. And here you can see the overall mean theta power of each pathway. The Shaffer collateral it has the lowest theta power, so you could say that, okay, probably the reason why it's the least synchronized with the rest of them is just because it's not oscillatory by itself. But if you look at other pathways like the entorhinal cortex layer 3 and layer 2, that they are also very close in their theta spectra with the Shaffer collateral, they seem to be much more dependent with each other and with the rest of the network than the Shaffer collateral. So this is this result looks a bit like the previous slide. So to conclude, yes. To conclude, the novelty of this year's work is the fact that we applied the ICA in recordings from the entorhinal cortex, which I haven't seen it in the literature. There are some preliminary results showing that the Shaffer collateral seems to be the one driving the dynamics, the synchronization. And to do, there are a lot of stuff to do actually, but most important, I would say we have to perform this analysis for more animals because results from just one animal is nothing in experimental neuroscience. But what I want to do, and I find it more interesting, is also to see a little bit the directionality of the network. So use mutual information and this kind of metrics in order to try and see whether the synchronization between two pathways affects the synchronization in the future between other pathways. And here we are also very open to suggestions from people at the FISC that know more stuff than probably me. Thank you very much, and thanks again to my supervisors of the thesis. And now is the time of questions. Let's start with the questions here, and then we can go into.
Thank you. Questions? I have one. When you show your phase calculation as compared to the go back, no, back. Go back.
From the literature, no?
Yeah, well, the literature and the one you computed, no, before. What was it that the one you estimated from the hippocampus to the entorhinal cortex that you have the.
This one, no?
Yes, that one. You say that the right side would be entorhinal cortex layer 2, close to the middle would be entorhinal cortex, okay, layer 3 and layer 2, that in principle are separated by pi, 180 degrees, and you don't find that. You find around 90.
Exactly.
But then you get 180 in the hippocampus. Is this contradictory to what you showed before? Because before you showed that they were clearly in antiphase.
So, yes. So, yes, of course. The problem is that here we see a complete reversal. So from peak to bottom, which is 180, while what we see here is 90 degrees. And the answer is that probably they don't reach the entorhinal cortex layer 1. So if you notice here, it's a continuous transition. So this is the layer 3. Here at layer 2, we are in the middle. So this would be the 90 degrees. And then we reach layer 1 if you go deeper. And I have done it, this kind of, I have performed this analysis for all the animals, and we never reached to 180. And probably it's just because the way that you pierce through the entorhinal cortex, you can't really reach the entorhinal cortex layer 1. The hippocampus is a really nice layer because you can pierce it in a way that you can gather information from every layer. The entorhinal cortex, it bends a little bit, so it's much more complicated and is much more mixed. So yeah.
Okay, thank you. Any other question?
So we are using. So we have 8 animals, 5 of them are for team maze. Some types of analysis I have done it for all of them. Some types of analysis I have done it for one of them. So this thing I have done it for all of them. I'm not showing you here. I'm going to be very honest with you. I'm showing you the best one. There are much worse in terms of what you expect. So here you can find more weird stuff at the entorhinal cortex. The hippocampus is always very stereotypical. But the entorhinal cortex, there are some animals they do weird stuff. Have I answered? Okay.
Any other question?
No, so we can go to online. I think Leo.
Yeah, really very exciting results. Thank you.
Wait a bit, we can't hear you.
Can you hear me? Can you hear me? Hello? Hi? Can you
Okay,
hear me?
say it. I'm going to come to the here.
Can you hear me?
Yes.
Okay, so yeah, no, I was just saying that it was really exciting results. Thanks for that. And I was just wondering if you have other data sets to compare with this in a sense of different behaviors or just not doing anything, or your data set is always when they are doing the same experiment. That could be a control, and that we could look at different things as well in this control. But I was just wondering what type of data set you have with this experimental recording.
We also have asked some people that they are very famous in the field for some of their data. I have done some type of analysis, but the problem with each data set is that it has its own peculiarity. Unfortunately, in the field, we are not very organized in how we report things. I have not done this kind of analysis. Like, for example, I have not generated this picture for data sets of different labs, even though we have from one different lab, just because I can't understand what I am watching at. I think that they have not ordered their recording sites from the animals or whatever. The problem is that they are not standardized. This data set has different tasks. Mainly, there is the team maze, as I said, which is 4 animals, or five animals, and then 3 animals, they are just running in a circular maze, so they don't have this decision part of being in the center. That's why I haven't showed it here. And also we have resting states. So the animal is just in its cage. Sometimes it's sleeping just by the fact that it's not moving. Sometimes it's just around in its cage. But we haven't really looked on the synchronization there either. Okay. Is there any other question from online? No? So, okay.
Thank you.
All right.